will talk about a new functional proteomics approach developed by her lab. Paola, please.
Thank you very much also for inviting me. And I also want to thank Yvette and her colleagues for the wonderful tour we did yesterday of your laboratories. It was a virtual tour, but it felt so real. And I got very excited about your technologies. So thank you for that.
Thank you, it was a pleasure.
So let's move on to the talk now. So with this presentation, I would like to propose a new concept. And this concept is that protein structural changes can serve as a new omic or global readout to detect functional alterations and to generate testable molecular hypotheses. So that's the concept, and I will now explain what I mean by this. So when we are interested in studying a complex phenotype, for example, the responses of cells to a perturbation or a disease, and when we want to use an omic approach, we typically want to first understand which portions of a cellular network respond to the perturbation. And when the components of these networks are proteins, we can use mass spectrometry-based proteomics for that, as you certainly know. When we apply these approaches, we can generate lists of proteins that change in abundance under the condition of interest relative to the control condition. And then what is typically done, starting from these lists, is some exploratory analysis. For example, clustering analysis to identify groups of proteins that behave the same. For example, proteins that go up or proteins that go down. And then also very common is to perform some kind of functional enrichment analysis to understand if proteins within the same cluster have anything in common. For example, if they are associated to the same biological process or pathway. And if we find something enriched there, then we typically conclude that these modules are somehow involved in the phenotype we are interested in. So this approach has been used a lot in the literature, very successfully. Also, my group used it in a number of studies. However, we all realize that it has some limitations. And the first limitation is that it's typically not easy to generate molecular hypotheses just out of a list of regulated proteins. So it's very difficult to try and imagine what could be going on at the molecular level based on a list of proteins and their fold changes. The second problem is that the abundances of proteins don't necessarily change when pathways are activated in a cell. And I would like to make an example of this. This is a pathway which is activated in yeast cells under conditions of osmotic stress on a very short time scale, for example, less than 10 minutes. It starts here with a cascade of phosphorylation events. These are MAP kinases, for those of you who are familiar with this. And then this triggers protein-protein interactions and protein-DNA interactions. Downstream of this, a metabolite is produced, which binds here to an enzyme and increases the activity of this enzyme. The net effect is that fluxes are redirected away from the production of pyruvate and into the production of glycerol. So all that happens here for all the enzymes you see associated to these reactions is a decrease in the concentrations of substrates. So none of these events would be captured by a classical proteomic analysis, protein expression analysis, because the levels of all the proteins you see here remain exactly the same when this pathway is activated. So the question I would like to ask with my presentation is there a way to capture these various types of molecular events which occur almost simultaneously in a cell within the same analysis? Can we do that? Do they have anything in common? So if you think about it, there is something they have in common. Think about post-translational modifications, binding of small molecules, protein-protein interactions, protein-DNA interactions, and also two events I did not talk about, aggregation events and cleavages. They all have something in common. They all locally or globally affect the structure of the proteins involved. So what I would like to propose is that by monitoring structural states of proteins, we might be able to detect altered functional states on a global scale. That's the concept. So how can we probe alterations in protein structural states? A few years ago, as Yvette mentioned, my group developed a method which we called limited proteolysis coupled to mass spectrometry. It's based on a relatively old concept, the principle of limited proteolysis, and we modified it to enable its application on a proteome-wide scale. So the idea is that we apply a non-specific protease, or better, low specificity protease. We like very much proteinase K. We apply it for a short time to a protein substrate. And then we quickly quench the reaction, for example, after 5 minutes. Under these conditions, the protease will cleave the most flexible and accessible regions in a protein, because this makes the reaction thermodynamically favorable. So the cleavages will be structure specific. When the structure of the protein changes, this reaction will produce different fragments. So we generate structure-specific fragments, but these fragments are still a bit too large for bottom-up proteomics. So what we do next is to denature them with guanidinium or urea, and then we digest them completely with trypsin. So from here on, that's the classical proteomics approach you're all familiar with. And then we measure the resulting peptides by mass spectrometry. Bottom line is that we generate proteolytic fragments that change when the structure of a protein changes, and we can measure these proteolytic fingerprints for all detectable proteins in a lysate. Then we convert these proteolytic fingerprints into what we call now structural barcodes. The name is fancy, but it's a pretty simple concept again. We basically plot the intensities of our peptides along the sequence of the protein we are interested in. And then when we apply a perturbation to our cells, the barcodes tell you which proteins remained structurally invariant and which proteins underwent a structural change. For example, in this case, you see the protein here with the orange bars. So this is the structurally altered protein. And based on the position of the bars, we can tell where exactly the structural change occurred. Now we say with a resolution of around 10 amino acids, because that's the average length of the peptides we measure. So when we started developing this approach back in 2013, we tested it first on a pretty large conformational change of a protein. That's the process of protein aggregation and amyloid formation. So we focused on a protein called alpha-synuclein. I'm sure several of you have heard about this. This is a protein involved in Parkinson's disease. Under normal conditions, it's thought to be a natively unfolded protein, so disordered, and then when Parkinson's disease develops, it's known to form aggregates in the brain. These aggregates, so these are basically filaments or fibrils, fibrillar structures, rich in beta sheets. So what we did back then was to start from the pure recombinant synuclein preparation, and then we let alpha-synuclein polymerize into amyloid fibrils. And then we spiked either the unfolded form or the amyloid form of alpha-synuclein into cell extracts. So these were two replicates of a yeast cell extract, and we chose yeast because it does not contain alpha-synuclein. And then we asked if we could capture this small difference between the two samples, basically an altered conformational state of one single protein out of about 4,000 expressed proteins in this case. These are the results we saw back then. So we saw that two peptides, the green peptides you see here, were only detectable when we had monomeric alpha-synuclein in the lysate, while when we had the amyloid version of alpha-synuclein, the green peptides were gone and the blue peptides appeared instead. So what are these peptides? The blue peptides map to the amyloid core of alpha-synuclein. It's these two peptides you see here. I hope you see my cursor. And so when the aggregate is formed, proteinase K cannot cleave anywhere here in this region. And so these peptides are only produced after denaturation and tryptic digestion. So these two are classical tryptic peptides, as you would see in a normal proteomic experiment. Now, when the protein is instead monomeric and unfolded, proteinase K can cleave here and also here. So the blue peptides are gone, and their smaller fragments appear. So from here to here, and from here to here. And these are the green peptides you see here. So this is pretty much the first experiment we did, and I still show it because it was really a turning point for us. When we saw this picture, we realized that we had now markers to probe the conformational conversions of alpha-synuclein directly in a total cell extract. So this was 2013-14, and that's the picture in 2016. So we identified a few more fragments that capture the conformational transitions of alpha-synuclein from the N-terminus to the C-terminus. It's in total about 60 peptides. And then that's the picture today. So we further expanded the coverage of alpha-synuclein, and we can detect a large set of peptides, fully tryptic peptides, half tryptic peptides, again, to capture subtle structural transitions of this protein. And we can now distinguish not only the two extreme conformational states of alpha-synuclein, so the completely disordered state and the amyloid state, but also three more structural states of alpha-synuclein that have been characterized in vitro. That's a principal component analysis, just to show you that the fingerprints can separate or differentiate these structures. And now what comes next? Now we are using these fingerprints to characterize the in situ structures of alpha-synuclein. Basically, we are looking into cells that express or overexpress alpha-synuclein and into tissues and try to match fingerprints collected in vitro from the pure protein preparations with fingerprints we collect in situ from cell and tissue extracts to try and understand which of these structures of alpha-synuclein occurs in vivo. So these were the first experiments, and then we asked, does this approach work only for very pronounced structural changes, like the process of amyloid conversion, or does it work also for more subtle events? And we directly asked if we could capture binding events between proteins and small molecules. Our idea was, if a small molecule binds a protein, maybe it triggers a conformational change, maybe not, it just binds there, it sticks to the protein, but still it should hinder proteolytic digestion or accessibility at the binding site. So minimally, we would expect to see a change at the binding site. This was the hypothesis. This idea with experiments in which we lysed cells, yeast or mammalian cells, we extracted proteins under native conditions, and then we added a small molecule to the extract. And the idea was we should capture a change in the structural barcodes of proteins that directly bind the metabolite or the drug. And I will now show you also here the first experiment we did at that time. We focused on a drug called cerulenin, which is known to bind a single protein in yeast, the protein fatty acid synthase subunit 2. So we lysed yeast cells, and then we added cerulenin, which is an antifungal compound, by the way. It kills yeast. So we added cerulenin to the lysate, and then we asked which proteins change their proteolytic patterns. And we detected here only one protein with altered peptide fingerprints out of more than 2,500 proteins we measured in this experiment. And this protein was fatty acid synthase, subunit 2. And then the surprise came when we asked which peptide changed in this experiment, because a single peptide changed, and this was exactly at the binding site of cerulenin. We then repeated this type of experiment with a number of small molecules. It doesn't work always this well. Sometimes we see changes at the binding site, but also somewhere else. And this could indicate that there are also conformational changes induced by binding of the small molecule. So in those cases, it's of course harder to tell which is the binding site. But in a number of cases, we do see only peptides at the binding site. So we thought of exploiting this approach, and we focused on the detection of protein metabolite interactions, because these interactions are known to profoundly affect the activities of enzymes, but they are very difficult to identify. And most of the ones we know have been discovered by in vitro experiments where people incubated a protein with a metabolite based on a hypothesis and then probed binding by activity assays or binding assays in vitro. So we thought maybe we can use this to detect these interactions on a large scale. We focused on about 20 metabolites. These are the ones you see here in orange. And then we added each of these metabolites to lysates of E. coli cells. So this is work done in bacteria. And then for each of these metabolites, we produced one of these volcano plots. So these are proteins that undergo some kind of structural alteration when a metabolite is added to the lysate. Some metabolites were very specific. For example, this is the volcano plot for phenylalanine. Some were very promiscuous. For example, this is the plot, I believe, for ATP or GTP. And in total, we detected 1,600 or so interactions, of which 1,400 were new and about 200 something were known. And then we had the idea of using the position of the binding site to try and classify interaction as putative catalytic interactions. So these are interactions occurring at known active sites. So for example, between an enzyme and the substrate, or an enzyme and the product of a reaction. And regulatory interactions, so any interaction occurring far away from an active site. And then we reverted to in vitro assays and validated about 20 of these interactions. So what did we learn out of this study? We learned that metabolite binding proteins cover a broad range of functions. They are not only restricted to metabolism. However, within metabolism, metabolite binding proteins preferentially occur in the subnetwork involving the production step of the metabolite. For example, this is phosphoenol pyruvate. This is where PEP is produced, and we detected interactions mostly upstream and downstream of the PEP biosynthesis step. We also looked into core proteins. So core proteins are proteins that never change in abundance, whatever you do to E. coli cells. Someone looked into that, that's the group of Bernhard Paulson. They came up with a group of proteins that are just incredibly stable across more than 300 perturbations of E. coli cells. And we found that these proteins were enriched in metabolite binding proteins, suggesting that these proteins are transcriptionally stable. However, they respond to perturbations by binding of metabolites. We also looked into the concept of enzyme promiscuity. Basically, we looked into those proteins that bound many metabolites based on our data. And we found that most of these interactions occurred at known active sites, suggesting that enzyme promiscuity is largely due to substrate ambiguity. So meaning that enzymes can just catalyze more reactions than we initially thought. So out of these experiments, we learned that this approach can capture protein small molecule interactions. And then a question I started to get all the time when I presented this data was, does it also work to detect protein-protein interactions? So now we tested this, and we focused on an interaction between the protein GAPDH, glyceraldehyde phosphate dehydrogenase, and an antibody binding this protein or targeting this protein, using as a control an antibody anti-rapid. So when we added this antibody to GAPDH, only 3 peptides out of the big structure of GAPDH changed, and these 3 peptides were all located at the known epitope region of GAPDH, suggesting that indeed, like for small molecules, we can capture here binding site. And I have to say, I even have the impression that for proteins it works better than for small molecules, simply because the interaction surface is larger, so you have a better chance to detect peptides right there. And next, we asked whether it also works for membrane proteins. And here we focused on, again, a known interaction between a gigantic transmembrane protein, that's the protein adenylyl cyclase 8, and calmodulin. So this is a known interaction, and it's known to occur here, where you see the blue rectangles. So these are the calmodulin binding sites of adenylyl cyclase 8. We worked here with a crude membrane prep. So basically we lysed the hex cells, and then we spun down membranes, resuspended them, and then added to the membrane suspension calmodulin at different concentrations. We measured then peptides after the limited proteolysis and trypsin digestion steps. And here you see the coverage of adenylyl cyclase. So in blue, you see peptides we measured. We did not capture the transmembrane regions, unfortunately, but that's a typical problem in proteomics. But the coverage of the protein was in general pretty good. And then of these peptides, only 3 changed upon addition of calmodulin and proportionally to the amount of calmodulin we added. And these 3 peptides you see down here were again located at the known binding sites of calmodulin, 1 here and the other 2 down here. So again, these are very preliminary data. It's the only 2 experiments we did a month ago, but I think it's quite promising. So to summarize this part, we learned from these experiments that using this approach, we can detect protein conformational changes and binding events. We can also detect aggregation events, as I've shown for alpha-synuclein. And something I did not talk about is that you can also use this technique to probe the unfolding of proteins after addition of a certain denaturant. For example, when you heat up a lysate or when you add urea, you can monitor the unfolding profile of proteins in a lysate using this approach. But now let's go back to the question I posed at the very beginning of my talk, and this was, can a global structural analysis simultaneously capture different types of molecular events? So far, I've shown you that we can detect individual or single types of events, aggregation or binding. And now the question is, can we detect all of them simultaneously? So I would like to go back to the pathway I've shown you before, the one which is activated in yeast under conditions of osmotic stress. And the question was, can we capture all the different types of events you see here? So to address that, we grew yeast cells under conditions of osmotic stress. And again, for a short time, in this case, 10 minutes. In parallel, we also grew yeast under conditions of heat shock to assess the specificity of our readout. And then we measured in parallel altered structural states and protein abundance changes using a classical proteomic approach. So what you see here in this histogram is the number of proteins that change in abundance in green and in terms of structure in orange. So first, not many proteins change their abundance, and that's expected because the time scale is very short, only 10 minutes. But many more proteins change their structure. Then we performed the Geo Enrichment Analysis, and here the results are shown as a gray rectangle, and the color scale indicates the Q value, so the enrichment of a given Geo term you see on the right side. So using protein abundance data, we didn't detect a lot of Geo terms, just the 3 down here, and that's again expected because the number of proteins that changes is very, very small. When we looked at protein structural data, we captured a certain number of Geo terms. These especially were enriched under conditions of heat shock, these for osmotic stress, and the ones you see at the bottom here in both conditions. And these terms boil down to many of the known suspects. So for heat shock, we detected terms that were linked to protein folding, the formation of stress granules, protein biosynthesis, and the regulation of translation, and this is known to be regulated under conditions of heat shock. Under the osmotic stress condition, we detect different Geo terms, for example, glycerol metabolism and glycolysis, and that's the pathway I've shown you on the previous slide. Then the response to salt stress, which is connected to that, and fatty acid beta oxidation. Now let's go back to the pathway we were interested in. Of all the proteins you see here, the ones in orange are the ones that underwent a structural change based on the LIP readout. These 2 you see here in gray we did not capture. And again, the surprise came when we looked at where these structural alterations occurred. So what you see here is the structural barcodes. So basically peptides mapping to the sequence of each protein and along the sequence of the protein. And in orange, you see the regions that undergo a structural change. And I say it was a surprise because we realized that the changes were extremely localized to only 1 or a couple of spots in a protein sequence. But then we understood what was going on when we mapped this data to available structures of these proteins. In the case of phosphorylation events, we typically capture the peptide at the phosphorylation site, sometimes a second peptide at the active site of those enzymes, suggesting that we might capture phosphorylation-induced activation of the enzyme. For enzymes that bound metabolites or small molecules, we captured peptides at the allosteric binding site. And down here, that was particularly unexpected. So these are all enzymes for which not a lot happens, only the concentrations of substrates change as a result of the redirection of fluxes. And here we mostly captured 1 or 2 peptides at known active sites. So we speculated that we might be capturing here changes in the occupancy of active sites, so having more or less a substrate bound to an enzyme. So we added specific substrates to the lysates of cells, and then we monitored the intensities of peptides at active sites against the concentration of the substrate we added. And the curves we obtained were very much reminiscent of a binding curve, suggesting that what we are capturing here is probably changes in the occupancy of active sites. So to summarize, using this approach, we can capture different types of molecular events occurring simultaneously with the resolution of single functional sites. And then to understand what is going on, you can try and map your data to the sequences or the structures of proteins to then understand which sites are specifically regulated. So these were events occurring on a short time scale where protein abundances don't change a lot. What about events occurring on longer time scales where protein abundances are also changing? So to understand what would happen, we grew E. coli cells in 8 different nutrient conditions. You see them here. And we chose these conditions because the group of Uwe Sauer, so that's the group next door to us here at the Institute of Molecular Systems Biology, so they had generated flux data and metabolite concentration data exactly under these conditions. So we thought this might be very interesting to understand what our approach probes. So we grew E. coli cells in these conditions, and then we measured in parallel protein abundance changes again and the protein structural changes. This is the result. So many more proteins undergo abundance changes under these conditions, and a few more undergo structural changes. That's the Geo Enrichment Analysis. And what was interesting here is that we did capture more terms from the protein abundance data, and that the Geo terms we captured were somewhat complementary when we compared the protein structure and protein abundance data, suggesting that these two readouts capture different types of biological events. But now back to the question, can structural changes probe functional changes on a large scale? How can we assess that? So here we had the idea of using fluxes for that, because fluxes through a reaction catalyzed by an enzyme change when something happens with that enzyme. For example, when the enzyme is phosphorylated and activity changes, or when the enzyme is allosterically regulated, or when the concentration of substrates change. So we thought we can compare flux and lip data to address the question you see on this slide. So we mapped all our data to central carbon metabolism in E. coli cells, for which we had also flux data. In orange, you see proteins that change their structure. In green, you see proteins that change their abundance, and the thickness of the reactions here is proportional to the change in flux the Sauer group measured for this particular reaction. So we compared all this data, and the bottom line is that 92 to 97% of enzymes that change the flux also change the lip pattern, suggesting that altered structures are a good readout for altered functional states, while only 30% of flux changes were explained by a change in the abundance of the respective enzymes. And this was somewhat known already in the field of metabolic regulation. And last, we also asked if we could capture new regulatory events using this approach, especially metabolite binding events, because we had an interest in that. So the data I've shown you before from E. coli were in vitro data, where we added metabolites to the lysates of E. coli cells and probed alterations in the lysate. Here we looked for metabolite binding events occurring in vivo. So we reasoned that if a metabolite binds a protein, we should in principle see a correlation between the level of that metabolite and the intensity of our lip peptide at the binding site. So we plotted the correlation of all metabolites against all proteins, and we found a few cases, about 80 of cases of, let's say, pairs of metabolites and proteins for which we found a good correlation. And you see them here. Some of these were known. And then we picked one for validation. And this is the interaction between the metabolite fructose bisphosphate and the protein PTSI. So fructose bisphosphate is produced here. It's an intermediate of glycolysis. And the protein PTSI is here. It's part of a phosphotransferase complex that internalizes glucose into E. coli cells and at the same time converts it to glucose 6-phosphate. So it's an enzyme, the protein we picked here. So we looked at which peptides correlate with the concentration of Fbp. And this is two peptides, the ones you see up here, and both peptides were located at the active site of this enzyme. So we speculated that Fbp might bound the active site of PTSI and act as a competitive inhibitor of the enzyme. So we performed molecular dynamic simulations from which we learned that indeed Fbp fits well into the active site of PTSI and it's pretty stable in there. So could act as a competitive inhibitor. And then we performed activity assays in vitro, adding Fbp to purified PTSI, and that's what we detected. So addition of Fbp resulted in a decrease in the activity of the enzyme, confirming that indeed Fbp inhibits the activity of PTSI. And this interaction actually makes a lot of biological sense because, as I told you, Fbp is produced here. It's an intermediate of glycolysis. So when glycolysis runs full power, a lot of Fbp is produced. So it makes sense that the cell might want to stop the further internalization of glucose to prevent accumulation of glycolytic intermediates. And this is the last experiments I wanted to show you. So to summarize, we believe that protein structures integrate different types of molecular events that affect protein function. That's why detecting altered structural states of proteins enables detecting altered functional states of proteins on a global scale. And now we suggest the integration of structural proteomics data into functional proteomics screens. And we believe that this will increase the capability to capture physiological and pathological alterations. I also described a new workflow to directly derive testable molecular hypotheses. If you think about the case of the interaction between PTSI and Fbp, so they are knowing the exact location of the alteration, so that's the active site of PTSI, enable the generation of a molecular hypothesis, a possibility that Fbp inhibits activity of this enzyme. And with this, I'm done, and I would like to thank people who contributed to these studies. First of all, Valentina Cappelletti, who led the study I just presented, together with Ilaria Piazza and Thomas Hauser, who left the lab. Alesh Holfeld and Dina Schuster generated the protein-protein interaction data. Lily Malinowska worked on alpha-synuclein, and Johan Feng is the PhD student who started the whole LipMS business back in 2013. So I feel like thanking him because without him we would not be here. These are our collaborators and these are our funding sources. And I thank you all very much for your attention.
Thank you so much, Paola. It's a wonderful presentation. It's really exciting to see how you are using mass spectrometry beyond the traditional method of just identifying and quantifying sequence, right? So how you use it for structural studies, it's really impressive. So now we are open for questions. Please, if anyone has questions, feel free to unmute yourself and ask.
Hello, can I ask a question?
Yes, please, go ahead.
This is Margaret Chung. Oh, this is a fantastic talk. It's really blown away. You mentioned about the time scale with regard to change of abundance and change in structure. How short is the time scale that you are looking at? Like you said, waiting a certain period that these two features did not really affect one another. So how short is short in your case? Thank you.
So thanks for your question. So the first data set I discussed was the osmotic stress data set where the time scale was about 10 minutes. Heat shock was 3 minutes. So this is really short. Maybe you can go even to shorter time scales, but this we haven't tried. So under those conditions, protein abundances didn't change a lot. longer time scale, in that case, we simply grew E. coli cells under different nutrient conditions. So they had all the time to adjust to the new nutrient condition. And then your question was, how do you disentangle protein abundance changes and protein structural changes? So there we use a simple trick. So we measure always in parallel also protein abundance changes. We always have a control sample where we don't add proteinase K. So that's a classical trypsin digest. So we measure protein abundances based on that, and then we apply a normalization factor. So we correct our proteolytic patterns, the ones that depend on proteinase K, we correct them for the change in protein abundance of the corresponding protein using the trypsin control. In this way, we can separate the two components. I hope this answered the question.
Thank you. Yes.
Stephen?
Yeah, absolutely fantastic talk. I want to run back to the laboratory and try it. So so one of the questions I have you looked at the impact on like adding growth factors or hormones?
No, I don't think we did anything in that direction. No, we haven't tried that.
So the second thing I have is on other cell types like mammalian cells, how do you extract the proteins without having in a native conformation without having trouble with lysosomal enzymes and other proteolytic enzymes in the mix?
Yeah, that's a great point, and we worked quite a bit there, and we made several mistakes as well. So we lyse cells mechanically first, not chemically. We don't add detergents, which means also that we lose more membrane proteins than in a classical proteomic approach. And then, yeah, so mostly mechanical lysis and omission of the detergents. You had a second question, which was?
That was in essence, how do you break mammalian cells up and not have the trouble with proteolytic enzymes?
Enzymes, yes. Okay, that was the second part of the question. Yeah, so we preserve structures by mechanical lysis, and then enzymes, we add now protease inhibitors. Mm-hmm. And we can add a lot of that because proteinase K is quite sturdy. It's not easily inhibited by protease inhibitors, and we add a lot of it. So that works. However, indeed, we break up lysosomes, the vacuoles, so we do release enzymes, and we do affect the subcellular localization of proteins, which might affect their structures. So that's something we cannot control.
Okay, thank you.
Yeah, I have a question on the same kind of realm. If you have intracellular proteolysis already, and when you're breaking your cells, how you tease apart which is intracellular proteolysis versus what is the result of your proteinase K.
Yeah, so if I got it right, you are asking about activity of endogenous proteases, so pre-existing cleavage events before we add proteinase K, right?
Yes, that's right.
Yeah, okay, so to correct for that, we again use the trypsin control, what we call the trypsin control. So the sample where we added only trypsin and not the combination of proteinase K and the trypsin, because you can compare the intensities of specific peptides, and if you see that the abundance of a tryptic peptide is already affected there, then you can speculate that this is due to the action of endogenous proteases. So you can do these comparisons at the peptide level to understand which are new cleavages and which were effects of pre-existing cleavages.
It makes sense, thank you.
Vlad has a question, I think.
Yeah, first, not a question, rather a comment or suggestion. There's a fundamental question in Parkinson's disease and Lewy body dementia. When there are Lewy bodies in the tissue present, is that the major component of alpha-synuclein? So if I do proteomic profiling of, say, substantia nigra for Parkinson with Lewy bodies, where is this alpha-synuclein going to be coming from? Is it in the aggregate state, or is it in normal, it's normal functioning state? And I think you're in a great position actually to answer this question, because we don't know. We see the changes and we don't know what they due to. Do they due to changes in normal alpha-synuclein or aggregated? And sometimes those changes kind of offset each other. So if you answer this question, that would be of real help for those of us working in neurodegenerative space.
Thank you. Yeah,
so
this is. Sorry.
Yeah, the other thing I noticed that using DIA label-free kind of single-shot analysis, have you combined it with TMT just to get a deeper coverage with fractionation, or there's something fundamental that precludes leap with
Feed is currently unavailable.
Not sure what that was. So yeah, so these are both great
When
points.
you have finished, please hang up or press the pound key for more options.
I'm not sure what's going on, but I don't know if you hear this too. So I answered the second question first, the DIA and TM versus DIA label-free versus TMT with fractionation. So we are trying this right now. There is nothing that prevents fractionating and labeling. So it was just laziness. We just tried first with the unfractionated samples and the DIA label-free because this was just the easiest way to measure the samples. But you're right, it would be worth trying. And then now to the Synuclein, that's a great point. That's actually why we started our analysis of the in vitro structures, exactly to then match them to in vivo state and see which of those structures are present. With the Parkinson's brains, there is a major caveat that the Lewy bodies are very small relative to the protein mass. So we tried that. We can detect alpha-synuclein, so that's great. But we detect a lot of alpha-synuclein in a physiological state, in a physiological structural state, because the percentage of sample that comes from the Lewy body is very small. So we compared even brains from Parkinson's patients and healthy controls, and we do see an increase in proteolytic protection of alpha-synuclein, but in the range of 5%, 10%. So that's borderline significant. That's why I think we need first to enrich for Lewy bodies to do this comparison more properly.
Okay, so it feels like majority of it is in normal physiological state.
That's what you see.
If I may ask the third question, so looks like your method is really good for picking up which protein binds a certain small molecule. So the alternative method is, as we know, thermal profiling. So can you comment on this and pros and cons of one method versus another?
Yeah. So as you can imagine, that's a question I receive frequently. And we even keep discussing this comparison with people who developed TPP. I'm in touch with Misha Savitsky, for example, and we often exchange thoughts on that. So I think that our approach provides structural information, therefore information on binding sites, so that's an advantage. A limitation is probably coverage. I think they have a larger coverage with a TPP approach because we need to detect exactly the binding site to identify an interaction, while with the TPP approach, you just need to detect one peptide per protein in principle to measure the melting curve. So we never really directly compare data, or maybe yes, in one study now recently we exchanged the data sets, and luckily the results were in good agreement. But I think that's my gut feeling. They have more sensitivity, and we provide information on the binding site.
Yeah, thank you, Paola. That's a good guidance.
Good.
Paola, I have a question. Wonderful work, first of all. So in your general rationale for this kind of approach is more like a hypothesis-driven, targeted approach. It's not a global discovery, right?
What I've shown was global, actually. So I come from a targeted proteomics field, from the SRM world, but what I've shown today is mostly unbiased screens, either based on shotgun or a DIA. So
because with the limited proteolysis and without the nature of protein, I would assume the coverage of protein would be limited, right? And this approach would be more powerful if you know what you try to find.
So you say the coverage is lower because with limited proteolysis you tend to capture unfolded regions, because that's a typical comment also. That's why you say the coverage is lower.
That's my assumption. Coverage will be lower compared to the typical bottom-up proteomics.
Yeah, so regarding the capability of the approach to capture only certain types of secondary structure, that's what we assumed at the very beginning. So we thought that probably with this approach we will have low coverage because it will only provide information on loops or unfolded regions. But in fact, then we started to see that LEAP datasets are not particularly enriched in unfolded regions. But when we look at lists of regulated proteins, we find proteins that are rich in beta sheets or alpha helices. And now we came to the conclusion that LEAP probes the change in probability of cleavage. So if a region transitions from an unfolded region to a helical state, we probe the change in the probability of digestion. But we can also detect changes from a helical state to a beta sheet state if these two types of secondary structure have a different accessibility to proteolysis. So in fact, we probe various types of structural transitions.
Interesting. Yeah, makes sense. Thank you.
Thank you. And these are all unbiased screens. Again, we measured by shotgun all the proteins we could detect.
Yeah.
Is anyone having any other question, please?
Yeah. Hi, Paola. This is Tao Lu. I have a question. So first of all, yeah, it's wonderful work that you're doing. It's really eye opener in the structural biology, and obviously, what can we go beyond, the protein expression, right? My question is more kind of a, I don't know, philosophical kind of a level, I guess. So in your mind, what is the biggest challenge for you to go even more effective in terms of probing more than just one type of, interaction? For instance, sometimes you have maybe, I don't know, I'm just assuming you have maybe overlapping events, and what kind of a technological advancement you wish you could have to make this, work even more effective?
Yeah, I think you hit a major problem of the method. Indeed, often there are simultaneous events affecting the same protein structure and coexisting conformational states. Obviously, we cannot disentangle those, and that's a problem. And we saw that in some studies. So it would be wonderful to be able to first separate proteins based on their structural states and then probe transitions with limited proteolysis to separate coexisting structural states. So that's one limitation. And then, of course, always increasing the coverage would be helpful for the reason I mentioned before, because here we cannot rely on a single peptide to identify a structural change. We need to probe exactly the region involved in the structural change. So if you have a binding event between a protein and a metabolite, the interaction interface is very small, and we need to capture exactly those peptides at the binding site. And so improving the coverage at the peptide level is crucial. And we tried a few things with little success, but now we have some new ideas on how to do that.
Thank you.
Yeah, so in this case, have you tried different enzymes besides the trypsin?
Beside trypsin, okay, so we tried many enzymes for the limited proteolysis step. So instead of proteinase K, we tried physing, elastase, subtilisin, thermolysin, and we also tried mixtures of proteases. So far, we always decided to move back to proteinase K and to stick to it because it's very aggressive, it's voracious, and it hits a variety of proteins. So we never obtain better results than with proteinase K. We also tried mixtures of proteases, hoping to probe a larger fraction of a proteome, but actually went the other way around. So we captured less cleavage sites because the complexity of the digests increased, and so the background, the proteomics background or the mass spec background increased as well. And so this obscured the detection of certain peptides. So now we don't use cocktails of proteases anymore.
Okay. I assume you identify the peptide, right? Always from like the highly abundant protein, right? So if that's the case, have you compared your method with the chemical cross-linking strategy? Some people claim they also can do in vivo, right? But in this case, also it's a highly abundant protein.
Yeah, that's right. So no, we didn't compare, so I wouldn't dare to make conclusions on the respective coverage. Yeah, it seems that at the moment it's still slightly lower, but I don't think the same system have been analyzed with both techniques. But I personally think that cross-linking is going exactly in that direction, and I would love to be able to combine lip with cross-linking, maybe surface labeling as well. So they are all techniques that can probe protein structural changes. I don't think the future is necessarily to doing lip analysis, but I believe in the power of detecting altered structures with whatever method. So probably cross-linking will get there very, very soon.
Yeah, thanks. Very nice work, I think. Yeah.
Thank you.
We have one more question from Steve, maybe, or Margaret, I think.
If I have, it's great. This is another questions about the philosophy, but not on the mass spec side, but for protein modeling side. If today, I saw that you use molecular dynamics simulations to help you to gain insights. What would you think that you will need to, if there's a capability that will help you in interpreting your results? What kind of computational aspect you think that will help?
Yeah, so having structures for a whole proteome will definitely improve interpretation of the data. This is not yet the case, but maybe with the recent improvements in predicting protein folds with AlphaFold and those approaches, maybe this will happen soon. So having 3D structures for proteins really helps, because then you can map the data to the structure and you can pinpoint with functional sites that are affected by a given perturbation, and that's very important. You can use sequence data if structures are not available. You can use information from UniProt, for example, to know if a region is involved in a post-translational modification or other things, but having structural data on top is even more powerful.
But isn't your emphasis is about alternated structures, and these protein predictions not necessarily predict the structures that's functional inside the cell? How would you reconcile the two?
Yeah, I see your point. Yeah, well, I don't know how to reconcile that. Yeah, but that's what we are doing now. So we take data from the PDB database, we take 3D structures from there, assuming that this structure at least generally recapitulate what happens in a cell. But your point is, in a cell the structure might be different. Yeah, this is true, and this we cannot. So we have no solution for that.
Thank you.
We have time for one more question, if anyone wants to ask.
Yeah, I think I'll ask this. One thing that struck me when you were talking about the protein structure, there's these new algorithms to very accurately predict protein structures from the primary sequence of the protein. I can see using that in your technique and thermal stability analysis to really start to put together a comprehensive idea of how these pathways work. So I was just, have you obviously you've thought of that.
Yeah, we would like to reach out to the AlphaFold people exactly to propose some kind of cooperation along these lines, so that, because they could look at structures on a large scale and we could probe structural alterations on a large scale. We started to do some analysis, so we are putting together now all the lip data we generated in the past 7-8 years, and we are looking into the most variable regions in a proteome. So across many perturbations, what are the regions that change the most? So we are starting to learn something, even in the absence of available structures. For example, unfolded regions, locally unfolded regions, vary the most in a proteome upon perturbations. And that's probably to be expected because these are the regions that maybe engage in different types of interactions. So, but that's where we are. This is early days for these types of analysis.
Yeah, and I'll just follow up. An obvious application for this, if you look at a drug that has an effect and you don't know what the drug is actually doing, because a lot of the drugs that are developed for diseases, they clearly don't actually work the way they think they work. Yeah. And so I would think this is really a great way of determining the site of action of some drugs or theoretical drugs.
Yeah. So after we published the metabolite protein interaction map, we received a large number of requests to perform target deconvolution analysis for a pharma company. So that's something we don't do to avoid becoming a service unit. So we outsource that. But indeed, this is interesting, but you can also do mechanism of action analysis. So if you add the drug to the lysate, you can capture the target, possibly. If you add the drug to intact cells, you can probe the pathways that are regulated. So you can use this approach in two ways and combine the results.
Yeah, very, very exciting work.
Thank you.
Okay, well, thank you so much again, Paola. This was really very exciting. Thank you everyone for attending and your questions. Just a reminder, tomorrow we have a series of informal discussions with Paola. If you are interested and you are not yet in the schedule, please contact me or Mary Marco so we can include you in the discussions.
And thank you also from me. This was a very inspiring and interactive Q&A. I really enjoyed it. Thank you all.
Thank you so much. We're looking forward to the discussions tomorrow.
See you tomorrow.
All right, thank you.
Thank you.
Everyone, thank you.
Bye-bye.